All right, so the first one I'm going to talk about is actually QUBES. And I'm going to go to QUBES, QUBES, and it'll fill in for me. But the URL is really qubeshub.org. And if I can find the chat here, I can put that there. So what QUBES is it's a platform for sharing curriculum and materials related to teaching. And there's also kind of a QUBES community, and there are groups that use QUBES to work together to develop curriculum. I found we used some of this in our January hackathon. It seemed a little bit overwhelming to introduce at the same time as we're learning all kinds of other new things. So we kind of postponed learning how to submit documents to QUBES, but now at least show you how to search for things. So we do intend to publish things that we produce when they're ready in QUBES. And we do have a group in QUBES called Antibody Engineers. If I click the little search thing and I type Antibody Engineers, we'll come to the Antibody Engineers group. You are all welcome to join this. This is where we put information about the group and about the hackathons. And I think if we went right back to that beginning page, I guess I thought I would have something about the hackathon here. This is, anyway, there will be, maybe I need to be logged in to see that, but you would see information about the hackathons, and I can message everybody when we're scheduling our next one and let you know how to sign up. Okay, if I go back to QUBES proper, oh, there we go. And I've just found it on the internet. You can also find some of the activities that we're developing there's only one that we have published on QUBES so far. And if you use the little search icon and you type in SARS, probably SARS-CoV-2 antibodies, yeah, here you go. You will get to the activity that we published in here, that the SARS-CoV-2 versus variants group is working on. And if I click this, I could just click download, and it would download all these files at once for me as a zip document, and I would have them all there. Also, alternatively, I could click any one of these and download any one of these and have that to work on or hand out to my students. Okay, so that is all accessible and all there. There may be a time when you have documents that you want to make sure people are logged in before they download. We will work out some of those details as we go. I'm told that you can do that. I'm not exactly sure how to do that yet. But the team leaders will be kind of responsible for gathering all these things together, and then we'll work with them to put them in things into QUBES and get semi-publications. QUBES also has some different statuses for publications. This was just a quick QUBES resource publication, but QUBES has an option to publish something as a peer-reviewed item, and we can also take things in QUBES and submit them to CourseSource. I wanted to wait on this one a little bit until we had actual students testing it, so we have a little bit more data before we do that, but that will be something that we will do sometime this fall or maybe early in the spring. Okay, and there's also lots of stuff in QUBES. I can't even begin to explain and describe every single thing in here. If you search for antibodies, you'll find all kinds of materials. You can see there's 23 different categories, 16 publications about antibodies, 4 blogs, 3 projects. So and that's just antibodies, I can look for any, a lot of it's ours, but we could look for any other kind of um, scientific topic, I think. Can I spell physics? It's probably physics. Yeah, physics in here too. Blogs, citations, publications. A lot of stuff. Okay, so that's QUBES. Are there questions about QUBES? Not sure I could answer them, but all right. So that's QUBES. The next thing I want to show you about is a database that Todd and I learned about earlier this summer called SabDab, which is the Structural Antibody Database. And I don't ever remember the URL. I just type SabDab and it will show up at the top of the Google page. So that's how I get there. If I look at what's in SabDab, I'm just going to the statistics page. SabDab contains over 6,400 different antibody structures, which is pretty amazing. And you can see about 5,000 of them were generated through X-ray diffraction, 1,365 through electron microscopy, and then we have a few by NMR, not terribly many. We also have information about structures where there's a paired heavy and light chain. That's something that's people have only been able to do very recently, because if you can imagine, it's much easier to sequence one mRNA and get that sequence than it is to get the other part, the part of the other chain that binds to that heavy chain or that light chain. We can also see we have a certain number of fragment variable regions. Of these 6,400 structures, about 4,900 have antigen in them too, so both antibody and the antigen, so you can see the interaction. And then we have some of these antibodies have affinity data, that is how tightly do they bind. And you can see the number of antibody structures that people have been solving is going up over time, which is interesting too. I think it's also kind of neat here at the bottom of the statistics page, they show you the kinds of things that these antibodies bind to. So the majority of them, right, 3,900 bind to proteins, but we've got quite a few binding to peptides, and then haptens, which are small chemical compounds that can generate an immune response, carbohydrates, nucleic acids, and then other things. As far as where do these antibodies come from, most of them come from humans. We have that kind of bias, apparently. But we've got a lot from mice, and then we've got some from llamas and things people have made, vicunas, combinations of human and mice antibodies, camels, macaques, rabbits, and a bunch of other things. So we have kind of a big species bias. Okay, but these are all the antibody structures that have been deposited in the public domain. So we've got a pretty good set to work from. If I want to find an antibody at SabDab, I can search for it by using a structure. I can search for a structure, I can search for a sequence. If I have the sequence of an antibody, let's see. This is kind of basically a blast search. It'll find a sequence for me. And if there's a structure in it, let me go back to the structures for just a minute. When I look for structures, you don't want to get all the structures because that would take a long time, but you can look for a specific PDB entry. You can look by some kind of attribute. That is, do you want to look by experimental method or type of antibody? If it's all of them or just a nanobody or some of the other small forms, by species, which is somewhat misleading in some ways, but we have information there. We can look for antibodies that bind to particular antigens, like hapten or nucleic acid or protein. We could look by whether it's a kappa or lambda light chain, if it has a constant region, if it does or not. And we can look for if it has an affinity value and some other information. These questions here about QUBES. QUBES is the coronavirus antibody database, which we can access here as well. And then there's another cool one called SabDab, which is a database of therapeutics. Okay, so let's say I have an antibody and I want to know about which parts of the antibody are thought to be binding to my antigen. That, in SabDab, is the option called a CDR search. Okay, and what I would do to find my CDR, I'm going to just get a specific structure here. I'm going to type 1 IgY, which is a mouse IgG. And I can look at this, and I think I can get the sequence out of here. Find out. It would tell me if there what kind of light chain I have, and if it has a constant region, which I already knew it did. And somewhere in here, pretty sure, maybe it's just in the summary file. I'm just opening up the summary file here, which I should have done in text instead of numbers. Okay. That was not a good choice. You know what? I will get, I'm going to do one other, I'm going to get a sequence. I'm going to go to NCBI and get a sequence for that antibody to show you what I want to see next. So 1 IgY. I want the protein sequence for the next part, and I know I can get it here. So I'm going to, it doesn't matter which one I would take. I'll just take this one. And I click, for me to get the protein sequence, I like to click the FASTA sequence. And I don't think I need the rest. So I just have the amino acid sequence of that and that heavy chain of the antibody. Okay, so SabDab will let me search and figure out where the complementarity determining regions are for that antibody. So I can put in my sequence. Oh, I guess I could have just put in the PDB ID. Ah, that would have been simple. We'll do that. Put in the PDB ID, get CDRs, and what it shows me, what I find from this, is I have the complementarity determining regions that are on the light chain. I have the complementarity determining regions that are on the heavy chain. And if I wanted to see that in IC and 3D, what I could do is I could select something here. I'll just pick, I'll pick H3 from this one. I could open up my structure in IC and 3D. And to see that particular complementarity region, I could paste the sequence up here at the top, the very top left-hand corner, up here, click search. And you can see it's highlighted that complementarity determining region in the antibody. Both the B chain, and I guess this must be the other, the D chain. So both those heavy chains. Okay, so other cool things with SabDab that we might do like I said, there's a therapeutic antibody database here, SabDab. And what you can do with SabDab is you can see different antibodies that are parts of drugs wherever those structures have been contributed to a database. So I could get immunotherapeutics. I could search for a specific therapeutic or by therapeutic by attribute. That is, what's the target for that therapeutic? I might, for example, maybe I want to know about antibodies that bind to SARS-CoV-2. I can keep searching, or I can just actually type an S and get there quicker. I could look for antibodies that bind to the spike protein or to the receptor binding domain, either one. Say get therapeutics. And it looks like I have 20 different antibodies that I could be investigating, which is kind of neat, right? If you're looking for things you might want to study, that this would be a good place to go. And it also shows me whether or not there's a structure. And in this column here, it shows me the most, oh wow, they updated it to August 2022, cool. It shows me where they are in the process of being approved as a treatment. So I can see this one at the top, amubarvimab is currently in phase 2 and 3 clinical trials. So that's kind of a handy thing. And I can see things that are been approved. I think, I'm not sure what TBC means, but I can also see if I scroll down, yeah, I guess I can just see if things have been approved or not. And NFD, no further development. I can also see if things are not being worked on anymore. So NFD, so this one, no further development. This one, no further development. So it gives me some things I might study with my students. One of the things that people might do with antibodies sometimes is if an antibody comes off patent, they can make a kind of drug called a biosimilar drug, which would be a drug that would have a similar activity to that antibody, but people would need to change it to make it different enough so that they could market it as a new drug. So this might be an antibody you might go if you were looking for things along those lines. Okay, so up here we also had the coronavirus antibody database. I'm not going to show you that, but I do want to show you some more of the resources here, more that the Oxford group has put together. And the one that I like here is SabDab, which is cool because they have some web apps that can do modeling for FV sequences. They can do side chain prediction, sequence numbering, which I'm not going to get into, or canonical form. But some two that I really like here are developability prediction and antibody humanization tool. Monica talked about how they might discover antibodies and then they have to change some of the sequences on the antibodies so that humans don't recognize the antibodies as being foreign. Because if I were to get injected with a mouse antibody, my body would say, mouse, 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 kill it, right? And try to destroy that antibody as quickly as possible. But if somebody had taken that sequence and replaced the mice sequences with human sequences, I might say, oh, that's an antibody. Cool. We'll just leave it alone and we will let it do its job. So people use this tool, this humanizing tool. They would put the sequence in, and we can just test that out with our mouse sequence here. I'll go back and get the protein sequence. If I take this and I put it into here, and I say submit, I got it. Oh, maybe I need the other part of it. Not sure if I needed the header part or not. You know what? We'll just get rid of all that and then put that in. Right. Okay. I don't know why I'm getting the error message. I'll have to go back and look at that a little bit later. But in theory, what we should get is we should get it should point out amino acids that are obviously not human. And then I could go through and I could do some modeling and use some of these modeling tools to figure out what I would need to do to change things. So the other thing I like in here with these prediction tools was back to SabDab Pred is the developability prediction tools. So what that is referring to are structural features in an antibody that might make it challenging to develop. And what this means is if you're a drug company or you're a biotech company and you want to make an antibody to use as a drug, you want to make sure it stays soluble. You don't want it ending up as this weird little precipitate at the bottom of a tube. You want to make sure that if there's anything that you can change that's going to increase the half-life of your antibody, that's an important thing to know. Any kind of feature that might make your antibody, might make it so you can't manufacture it, right? That's what the developability tools are going to pick out for you. So if we look at this, and we'll learn about what those features are once we click this. So we go to this one, and I put my sequence in, and I noticed it had this thing here. Not sure if I need this or not. I noticed that this one had a slash at the beginning. I might have missed having that slash with the other one. That might be something that it needs to have. I'm going to say submit and cross my fingers. Yeah. Okay, this one's going to work, I believe. So what it should give me is it should give me another internal server error.
Part of the error says the server might be overloaded.
Yeah. I'll try one more. And we'll see if that does it. If not, you may have to trust me that, yeah.
See, I think it might be overloaded, and so you have to come back later.
Okay. We'll come back later. It's a pretty cool resource. It is free and very powerful. And we don't know how much traffic it can actually handle. Okay. So the other thing I wanted to show you about today is next strain. So in the SARS-CoV-2 versus variant group, they are looking at evolution of SARS-CoV-2 and the ability of SARS-CoV-2 to escape from antibodies and whether or not there are changes in the virus that allow escape. One thing, but there's other things here at Next Strain as well. It's pretty neat. There are other pathogens that people are looking at. There's monkeypox sequences. There's an instance with influenza and Ebola and Yersinia pestis, which causes plague, and West Nile virus. And there's also many of these also have narratives like this does with West Nile virus, talking about how the phylogenetic data, what kind of story it's telling, and about where there have been outbreaks and how you look at, did people get this by traveling, all kinds of interesting things. Well, one thing I realized looking at the SARS-CoV-2 data last year was that the two different sections here had a little bit of different information about, as far as the data that you could get. One of these is using GSAID data, and it wasn't always easy to get the sequences. This other one that has open data you can actually get protein sequences from. And I have to say, I haven't looked at these others to see how true that is for the other instances and the other pathogens that people have looked at. But if I click this, what I can do is I can see a phylogenetic tree that starts from December 2019 and goes through the whole progress of the pandemic and shows me where different kinds of variants evolved and when they came about. I also have a map of the world, and up here in date range, if I were to pick play, I'm not going to go through the whole thing, I would see how the virus spread around the world, and I could see like when different variants started appearing, where they changed, and you can even see as it's kind of going across the tree here, it's mirrored on one side in the tree and mirrored on the other side in the map of the world. About this map, one thing I want to point out, and one thing that could be a little misleading about the phylogenetic tree and some of this data, is that the sizes of the circles here, they don't necessarily reflect all the cases where people have been sick. What they do reflect are the number of DNA sequences that were obtained and submitted to the database. And what you find is some countries are doing a lot of surveillance and a lot of sequencing, and other countries don't seem to be doing any, as far as we can tell, or at least not so much that it shows up as a significant amount. So you can see like which countries are really contributing to our knowledge base in understanding the dynamics and epidemiology of the virus. Okay, some of the other things we can see from this map, or from this dashboard page, is down here we have a map, a schematic that's a map of the genome of the virus. Each section, like ORF1A, maybe I can zoom in a little bit. ORF1A is one protein, ORF1B is another, S is the spike protein, and then we've got some other proteins here, the nucleocapsid, actually, I don't remember what that is, the matrix protein, I think it's maybe the capsid protein, but different proteins in the virus, and you can get an idea where mutations occur in the virus. So as you see here, we've got a really high, the height represents the frequency of mutations. We've got a really high frequency of mutations in this region right here, and that is the spike protein, which it makes sense, right? If the virus is evolving due to selection pressure from people's immune systems, or to be able to infect a wider variety of cells and more quickly, then that's what we see, evolution. We can also see frequencies of the different clades down below. So right now in August 2022, you can see that almost 80% are 22B of Omicron, and then we've got 6% or this other Omicron clade, and then 21L, this other Omicron clade. Viruses are defined as a clade, a group of viruses are defined as a clade if they're similar to a certain extent. Then once there are enough changes, once there's a certain number of changes, and I'm not sure what that number is, they call it another clade. So we get another name. Okay, if I were to, maybe I wanted to look at any one of these changes in the S protein, which is somewhat tricky because if I mouse over, my cursor gets stuck. Okay, so if I very carefully click one, there we go. Okay, I guess I zoomed in. Now I'll see if I can click one. Not always cooperative. Okay, so what I clicked right here is codon 371 in the S protein. You can see it's one that has a high frequency of mutation. And as I scroll down, I can see that almost all the sequences right now for that position have a phenylalanine at them because it's got an F, and F stands for phenylalanine. I can see that before that, there were some viruses that had an L there, which stands for leucine, and there were a few that had an S there, which stands for serine. But at this point, almost 100% of the sequences have a phenylalanine at that point. When I clicked that spot, that change, that selection was also reflected in both my map of the world. So I can see here what it's like in different parts of the world. And it also changed in my tree. And now interestingly, if I kind of zoom this in a little bit, carefully zoom it in a little bit, not always easy. There we go. You can see here, it's kind of just barely peeking out. There's some green things here. Okay, a green button or a green dot. And when I carefully hold my cursor there, I can see that this one, maybe I'll just click it. Okay, this one says that at that position, does it show me what I wanted to see? Now it's showing me a lot more. I'll go back. Okay, if I hold it there very carefully, it shows me that amino acid at S site, at the spike protein site 371, which would be amino acid, the position is 371. This one has a serine instead of all these others that have a phenylalanine. So looking back, apparently this is maybe a reversion or some kind of change. It was originally serine, way back here, changed to phenylalanine, and then now some of these are changing back to serine. It got kind of that green color. I know it's sort of hidden underneath the yellow dots, but there are some that are changing back. So yeah, we can see how things are changing over time. Some other kinds of things we can do. There's some filters over here that let us look at different kinds of information. I know we are looking at the SARS-CoV-2 database, but you can see here we also had dengue and Ebola and enterovirus and mumps and Zika, other things. We're looking at the open data. There's also data, we could limit it to different countries if we wanted, or regions of the world. And we can also change the way we color things. I had it, I'm going to reset this. Or maybe I won't reset it. Oh, there we go. I get rid of my filter. There. Well, if I have it by genotype at position 371 right now, but I could change it back to clade, which is what we were originally looking at. I could also, if I wanted to, change it to date or genotype or mutations or fitness or current frequency. But what I like, another one I like to look at is emerging lineages. So I'm no doubt you've heard you've probably in the news maybe heard of BA.5, BA.4, and BA 2.1. Okay, so you could look at sequences from those new lineages of Omicron that have been appearing. I can see them highlighted here. And now you might wonder, okay, well, what do those look like, right? What can I see about these? So to look at these sequences and find out information about them, I can click anyone, and it'll tell me this particular sequence is from July 20th, and it was a person in Japan. I can see who submitted it. As I scroll down, I can see that there's 27 mutations in the S1 protein, and the 27 reflects changes since the January 2020 version. So since the Wuhan fish market version. I could see the number of changes, nucleotide changes, and the number of amino acid changes in any one of the proteins. And what is really cool about looking at the open data is there's a link here that will allow me to go here and see that sequence in GenBank, which is very helpful. I will get way more than I want. In fact, I'll often get the whole entire genome. But I do have the option to look at a particular part of the sequence. If I select graphics, I've been looking at these enough that I remember where the S protein is. So it's not always so easy to see unless you're staring right at the computer, but here is ORF1, and this one must be another part of the ORF1 polyprotein. The green rectangles represent different proteins. The sequence underneath represents the green part represents the gene. The sequence underneath represents the protein. So what I could do with this is I could take this S protein from this BA.4, and I could either download the protein sequence, or I could get the, I could just copy the accession number, and I could use this in different ways. What I was doing with SARS-CoV-2 is I had a sequence of SARS-CoV-2, and let's see, I think it's 7KHM. No, maybe 7KMH. Yeah, there we go. I have a sequence of SARS-CoV-2 that has an antibody bound to it. I could open that up in ICN 3D and then take BLAST, go to Align Sequence to Structure, put that accession number into that box there, replace the default accession number with the spike protein that is in this structure. So it's 7KMH_C. Align with either BLAST or Smith-Waterman. I haven't found that the difference really matters that much. And I could see here how much the spike protein has changed from in this new sequence, how much has changed from the original sequence to how it is in this new sequence. And I could, even if I wanted to, which is I'm not going to do it all right now, but I'll just call up and show you where the antibody is. I think I could show all chains. Maybe that would do it. No. Well. There we go. So I could see how much the spike protein has changed and how those changes map to the portion of the spike protein that interacts with the antibody. And I can see, like, this is a part, the way the colors work, I don't know why. There we go. The way the colors work, red means it's completely the same. Light pink means it's pretty much the same. Blue and gray, they're going to be pretty different. So I can see there are some changes here that, you know, could very well impact antibody binding because it's changed quite a bit. And I'm not going to go through all of that right now. But you can look at the activity in QUBES and see what the SARS-CoV-2 versus antibody group is doing, and you will see all that. So that's my demo. Are there questions? Anything you want to see again? Totally overwhelming?
Sandra, I'm not sure if you can hear me, but that was very interesting. Thank you for sharing.
Thanks.
So, aren't you supposed to be out for dinner on your anniversary? So the
On our way back.
okay. I was gonna say the difference between the BLAST and Smith Waterman is primarily BLAST is gonna send data to the server and bring back the results, and the Smith Waterman happens in the app. So for local operation, and you don't want your data to go to NCBI, you can use the Smith Waterman.
Yeah, there's also some things you can do with IC&3D and looking at mutations so that you can kind of get even a better feeling for what the interactions are between the original amino acid and the antibody and the mutant amino acid and the antibody and how it's changed. So you get an idea that did this mutation allow the virus? Is this mutation going to allow the virus to escape, or does it not matter? Or is it not likely to matter? But now, when you read the news and you hear about the N501Y mutation or something, you can know, oh yeah, they're talking about the asparagine at 501 getting replaced by a tyrosine, for example. More questions? No more questions?